format of this series, again, we'll see how this works, is that the speaker has 20 minutes. And if you go over 1 minute, we'll be okay. But that's 20 minutes, roughly, after the introduction. And then we'll have Q&A, open discussion. And if you need to leave, this is not like your formal meeting. So if you need to leave, go work on an experiment. I think your PIs will be very happy about that. But so feel free. Again, this is just the chance to network, but you're welcome to take off just quietly, but then we'll stick around until 1 o'clock, and then, that's the format. Does that sound good? Okay, with that, let me introduce Dr. Todd Herron. Thank you for coming in at the last minute just a week ago for today's series. We're finalizing the framework. And Todd is a physiologist who did his PhD at the University of Missouri, Columbia. We can see his slide if you've got it, or the first page of his slide. And did a postdoctoral fellowship at King's College in London in molecular and cellular cardiology, postdoc at the University of Michigan. He's a cardiovascular scientist who specializes in human-induced stem cells and works with Joe Wu, who is the director of the Cardiovascular Institute and the really outstanding resource that Todd will tell us more about of pluripotent stem cells. More about Todd. Todd has a background in stem cell biology, cardiac electrophysiology, translational bioengineering. He's done preclinical drug screening, and this is really the theme of their group, which is using newer alternative methods to do drug discovery. And those who were part of the drug discovery symposium last week will know that Joe Wu was the main organizer, and so that's part of Todd's group. And Todd is affiliated with Greenstone Biosciences, which is literally right across the street, or down the street somewhere. Yeah, so you can go visit him along with his colleagues here, Hannet and Sure. Yeah. Right. So thank you. And today he'll be talking to us about patient-specific disease modeling and therapy discovery using 3D organoids. And so please join me in welcoming Todd Herron.
All right, thank you. I think I'll stay seated so you can see the screen. And as Sohail introduced, it's informal. So if there's any questions during the talk, please feel free to interrupt me, and I can help. I can try to answer any questions. I also want to just say, as Sohail introduced me, my expertise is in cardiac biology. So I'm a little bit overwhelmed being in the presence of so many experts in pancreatic physiology and pancreatic function. While I was at the University of Michigan.
I and maybe 3 or 4 others are pancreatic function. This is a broad audience.
Okay.
Of Arweh Hall.
Okay.
This domain.
Okay. So I do have some experience with using pancreatic organoids, and I'm very proud that I was part of a publication that came out in 2025 from my work at the University of Michigan, looking at some of the mitochondrial and metabolic function of stem cell-derived islets that are used for research. So today I'm going to talk mostly about induced pluripotent stem cells. So as most of you probably know, there are 2 different kinds of pluripotent stem cells. The original, or the old school, is embryonic stem cells, which are useful. But as you can see, in some states, using embryonic stem cells is a crime. Like in Alabama. So that's because an embryo must be destroyed in order to obtain the cells that are pluripotent. So the world, and especially Greenstone and Joe Wu's lab at Stanford, we prefer to utilize hiPSCs. So hiPSCs are human-induced pluripotent stem cells that are made from a patient's blood sample. So here, the other advantage of using iPSCs is that there's a patient who you can relate in vitro data to their clinical course. And you can recreate some genetic basis of disease in vitro utilizing patient-specific cells. And we think that it's also useful for patient-specific medication screening. So for many chronic diseases, there are unknown treatments that can be tested in vitro that can't be tested in the clinic. So we think it also can provide some clues or some evidence for where to start in a treatment with a patient. So some of the work that I'll show today relates to type 1 diabetes, but I'll also show some projects that we're working on now where we're studying pancreatitis and looking at the other function of the pancreas. So working with stem cells is a lot of hard work. It's very labor intensive, and that's because it takes a long time to progress from the pluripotent stem cells, which are kind of like the seeds of the project. So once we have generated the iPSCs for a patient, then basically we have that patient's cells forever, because the cells can be maintained forever as long as you take care of them properly. They can be frozen, they can be reanimated. And so it's a way to have that patient at the lab bench basically over and over and over and over again for repeated measures and for disease modeling as well as medication testing. So this is the process that we use. There's many different steps along the way, going from stem cells to pancreatic progenitors. Fortunately, there are commercial products and kits from a company called Stem Cell Technologies that we utilize. So these are very easy to follow, and with very high efficiency, we can generate high purity pancreatic progenitor cells. And then in some of the data that I'll show you, I've utilized these pancreatic progenitors to create stem cell derived islets that are made up of pancreatic beta cells, pancreatic alpha cells, as well as delta cells. And I'll show you that they also are functionally mature and they respond to stimulation with glucose by releasing insulin. And these are just some of the one of the leaders in the field whose protocols I follow is Timo Anankansky. He's from Europe, and he's really done a great job figuring out these differentiation approaches and differentiation techniques. So this is immunofluorescent results showing what the cells typically look like when we go from stem cells to getting the stage 4 pancreatic progenitor cells. Here you can see expression of NKX 6.1 at high abundance in the cells in this field of view, as well as PDX1, which is a molecular marker that tells you about the differentiation stage of the cells. And this shows that at stage 4, typically around 90% of the cells are expressing PDX1. So it's a very efficient protocol, very high purity of pancreatic progenitors. And also those cells, some of them express NKX 6.1, and then the ones that are positive for both is around 60% that are double positive for NKX 6.1 as well as PDX1. This just shows the reproducibility of this protocol. So this shows the results over the course of about a year in my time at the University of Michigan when we repeatedly perform these experiments. And you can see for each experiment when we measured the PDX1 population at stage 4 that it's consistently around 90% PDX1 positive cells. So it's very reproducible, very efficient, and it's a good workflow to produce the cells that we want to study for studying either type 1 diabetes or type 2 diabetes, as well as other pancreatic diseases like pancreatitis or pancreatic cancer. So this is more characterization of those cells, just showing the pancreatic progenitors. We can also mature them and coax them to become mature beta cells. So this is staining for C-peptide, which is an indication of the insulin content. And also, besides being present, this is confocal imaging. You can also see that it's very punctate in nature, indicating that it's stored in vesicles, as insulin is when it's ready to be released into the systemic circulation. This is looking at the alpha cells. So besides making beta cells, this protocol also generates alpha cells that express glucagon, shown here in green. So it's pretty interesting when we do this staining. It's always predominantly beta cells with a lower number of alpha cells and delta cells, which I think mimics what happens in vivo, where the islet is predominantly beta cells. So somehow the cells figure out how many of each cell type they need. I don't know how it happens, but the cells are very smart and kind of control the stoichiometry or the cell population, I think, based on what they need or what they need for survival. And I think people tease me sometimes, but I like to refer to Jurassic Park, where I think Jeff Goldblum's character said, life finds a way. Somehow the cells find a way to survive and to propagate and to stay alive. So this shows presence of the delta cells by probing for SST hormone, showing also that there's presence of the delta cells that are created in this differentiation process. So the main players of the pancreatic islet are all generated in this protocol. So then the next question that we had to kind of make a decision on in the workflow is how to make healthy organoids from these cells that we're generating. So it's really easy to generate these cells in 2D. So everything that I've shown you to now is all in two-dimensional cultures. But of course, the human body and organs are all three-dimensional. So now also with the new approach methodologies, there's also a big movement by the NIH and the FDA and the biomedical research field in general to move towards three-dimensional organoids as a more physiological approach than 2D monolayers.
Are these cells transplantable?
Transplantable. They can be transplanted. I haven't done any of those experiments, but at Greenstone, we do have a project where we've generated iPS cells where we've knocked out some of the immune response receptors so that the cells can be like stealth and not and kind of evade the native immune response. So that kind of cell type would probably be better for implantation.
Is derived from the same person, like patient derived iPS, then there should not be much of immune response.
That's the thought. Yeah, that's the theory. That's the hope of the iPS cells, that it can be autologous. I think right now the workflows kind of prevent that. So I think there's more of a movement by cell therapy companies to try to generate like a master cell line that could be applied to anyone, which sounds too good to be true. I think it's better if it's autologous.
For diabetes management, it will be very helpful because the cost of diabetes management for 10 years, 15 years will be much cheaper if we can transplant these beta cells.
Yeah. And there have been some, I don't know if they're clinical trials, but there has been some testing done in patients. And it's been shown that the implantation of these organoids does contribute to blood sugar regulation over a pretty long duration. I think I've seen some studies that have been followed up for up to a year. And the cells survive and they continue to contribute to blood sugar regulation. So yeah, definitely.
So to build on that point, the ongoing trial is run by Vertex Pharmaceuticals, and it's allogeneic islets. And they've had several successes in patients with very brittle diabetes. But those are not autologous, they're allogeneic. And they don't have the immunosuppression that ends up eventually destroying their kidneys. And since they're diabetic, that's not a good thing.
Yeah.
So an autologous approach using inducible stem cells would be very desirable. I'm not sure why Vertex went with the allogeneic off the shelf. Maybe because they used embryonic stem cells. I assume they did that.
Also marketability.
Yeah, so I mean, I think it's a workflow issue, whether you can get enough quickly enough.
Yeah, my dream, the ideal world is that every person would have iPSCs banked and just stored somewhere, so that when you need those cells for cell therapy or to test drugs, then at least 2 months of the process is completed. And then there would be an iPS cell line that you could go to, animate it, and get going on the work. I think that's probably the future of regenerative medicine. Yeah.
But isn't it also the case that some iPSC cell lines differentiate better into certain organs or into cells than others? So it might not be possible from every person to differentiate them into pancreatic cells. I mean, I know from my collaborators that some they can make endothelial cells well, and then some other lines don't make the endothelial cells so well. So you probably have to solve this issue as well, right?
Yeah, there is some of that, and I don't know how much that is inherent to the cell line or to the person doing the experiment or to other variables that might be at play. So, for example, most people utilize an extracellular matrix called matrigel to grow the cells, and that's an inherently variable reagent. Like from batch to batch, it's different. Even the way people handle it can cause variability. So there needs to be standardization of the protocols. And in my experience, if a cell is truly pluripotent, it can become any cell type. Sometimes if the iPSCs aren't handled properly, or a little bit of spontaneous differentiation occurs, that can also change the outcome. So there's a lot of, I think at the moment, there's a lot of person to person. When I say person to person, I mean laboratory to laboratory technician or scientific personnel that lead to the variability that you're talking about. I don't know that it's actually the cells as much as it is the whole approach.
Can I just add to that? There's a current transplantation technique called total pancreatectomy auto-islet cell transplantation, where patients with chronic pancreatitis, in which the organ is just extremely pain-inducing and intractable, require removal of the pancreas. And then there's a procedure where you try to salvage some of the remaining islets, and then you feed them back into the liver, and they implant in the liver. But I think it would be brilliant if you could augment those native islets with more autologous islets that are just generated from somebody's blood, prior to the procedure.
Yeah, there's a lot of possibilities with using the stem cells once the stem cells are made. All right, so everything I've shown you so far is in two-dimensional cell culture. So we found, I think I have some data slides maybe, the maturation of the islet cells to secrete insulin is always better when they're made into 3D organoids. So we've done glucose-stimulated insulin secretion, or GSIS, and 2D monolayers. And we see a little bit of insulin secretion, but when we take those same cells and put them into an organoid format, the amount of insulin secreted increases by like 2 to 3 times the total content, as well as what's released in response to glucose stimulation. So there are different ways to make organoids, and one very popular method used all over the world involves some kind of spinning. So here I've just shown 3 different platforms. One is a spinner flask, where there's basically a propeller inside that spins and makes the cells go around in an orbit and circles, which is effective to make organoids. Then there's also orbital shaking, where basically you put a plate or a flask of cells onto an orbital shaker, put it into the incubator, and just let them go around in orbit for a while inside the incubator. And then at the bottom, I think this is the best illustration of what happens inside the liquid, where there's a tornado. The reason I've shown this tornado is because all of these methods are a double-edged sword. So all of these methods, they do produce organoids, and the shear stress does induce some vascularization of the organoids, which is a good thing. But also that same shear stress causes damage to the organoids, where they become brittle, sometimes they fall apart, and sometimes their phenotype is determined by the shear stress. So sometimes you get an organoid, you get a mixture of healthy and damaged or sick organoids in the same batch. The other problem with all this orbital shaking that I've observed in my own laboratory and looking at the work of others is that they also produce highly variable diameters of organoids, where some are big, some are small, and some of them start even clumping together if you let them continue to spin. So what we started using is a new organoid plate production method developed by Corning. So this plate, basically, I've shown it here. So these flasks or cell culture plates have microwells engineered into the base of every plate. And in here, there are 12,000 microwells per plate. So it creates the potential to generate 12,000 organoids all at one time in one batch. And beyond this, we've started working with the Corning group. Also, they've created now what they call the 48K plate, which basically they've taken 4 of these and put them together, and then you can generate 48,000 organoids all at one time in one batch. And because the physical constraints of the microwells, the organoids are all very similar in diameter and very, very similar in shape and structure. And the other advantage of this approach is that it's easy to change the media. So with other approaches, like with the spinner flasks, when my technician would change media and then start spinning the cells again, we would just lose cells. So like we would start with a bunch of very good looking organoids, and then over the course of a week, we'd be down to like 50 organoids. And so I was like, we can't do anything with 50 organoids. It's not enough almost not enough to do like biochemical analysis. So when we started using these plates, it really made our lives easier and also increased the efficiency of good organoid production to where, and this was kind of a battle that I fought at the University of Michigan, is that the diabetes researchers there are very like stunned, like these aren't islets. These are some fictitious thing that you think is an islet. And whenever I made islets that were good enough that they started calling them islets, I'm done. They called these stem cell derived islets because they started to recognize that they're just as good as cadaver islets. So the other popular method for type 1 diabetes research is using cadaver islets, which learning from my colleagues at Michigan are also fraught with a lot of trouble, where you might get a whole batch from a patient, but then when they look for insulin content, there's like no insulin. Also, they would do the GSIS experiment in the cadaver islets and be very unsatisfied with the amount of insulin secreted. So when we were able to start routinely generating stem cell derived islets, they were very, very excited because now there was a renewable resource that was of high quality for their research. So then they didn't have to wonder, like, is this cadaver, they would almost be like crossing their fingers, like, please let these cadaver islets be good. And I was like, that's not good for science to be trying to get lucky, trying to be lucky in science. It should be like more systematic. So these plates are very good. We started using them. Each islet has basically its own home and facilitates media for a long term with very little loss of the organoids over time, because the organoids stay in their microwell while you change the media.
Excuse me, do they like bottom?
Sorry?
Do the organoid attach to the bottom?
The organoids at the bottom, yeah.
And that attached to the bottom? Like
No, it's a non-adherent plate.
So it's the media?
It's an ultra low attachment plate, so there's no attachment. The organoid's kind of suspended within the little chamber. In the chamber, Corning has better diagrams I can explain here, but I think it's like kind of like a dumbbell shape chamber, so that when you change media on top, the organoid is undisturbed.
Okay.
If you still have to be gentle, like if you go in there and just blast the media, yeah, they'll all come out, but you have to kind of be patient and control the speed of the pipetting so that you can change the media without disturbing the islets. And if you do disturb the islets, you can always spin them back down and then put them back into the plates, and then they'll fall back down into the microwells.
So compared to the spinning flask one, so what's the size difference between this smaller
So these ones are uniform in size. I think I'd have to do a measurement. They're between 50 to 100 microns in diameter. With the spinning, the diameter depends on how fast you spin. And the faster the rotation, generally the smaller the organoid. And then if you change the speed, then you can kind of change the size of the organoids based on the RPMs of the spinning flask. But I think that's also hard to reproduce, like batch to batch with the spinning, because there's many variables that go into the organoid formation besides just the spinning speed.
So the advantage to using those spinning flasks is like they introduce the flow, right? So
Right,
here
they
we
might
have a
introduce
static, like cultural, it's like no perfusion system here. So
In these, there's no perfusion. It's like a static culture where you change the media every 2 to 3 days.
so where is like a disadvantage compared to the
I haven't done that comparison. I don't know if functionally, I don't know if there's a difference between the two. I know structurally, just looking at the organoids, there's a difference.
because you said like the spinning flask, like because the flow, you can like increase the vascular reaching. So
It might
this one you
increase
don't have like the flow, so maybe you have less vascular.
Right. Yeah, I don't know if that's compared. Yeah.
Let me just make one announcement. It's 12:30. So those who would, feel like they would like to take off, they're welcome to. But and we'll keep this informal for the rest of the hour. So again, this is our time to connect, and please continue and have this informal discussion.
So this is a closer look at what those organoids look like in the microwells. So you can see they form nice tight boundaries. They're spherical in nature. And this is after about 18 days in the spheroid plate. So you can see that they stay there pretty good. There's loss of sun. Like here you can see an empty microwell, here an empty microwell. The other thing that I think is useful with this plate, so organoids are difficult to count using cell counting approaches. There's not really the best way to count the organoids, but here we use this as a way to count the organoids. So we know that if there's 100% coverage, then there's about 12,000 organoids. And if there's 50% coverage, we know that there's about 6,000 organoids. So as far as counting the organoids, we like to use this method also because it gives us a very close approximation of how many organoids we actually have. We do have an organoid cell counter, which is basically a cell counter that has some AI algorithm that detects organoids, but it still has the same problems as regular cell counting. With the other problem with counting the organoids is it's hard to dispense them evenly because gravity just starts to pull. Like when you're working with them, as soon as you spin them down and resuspend them, they start falling to the bottom very, very rapidly. The organoids, even though they are in their little microwells, it is easy to remove them. So when it's time to do your experiments, basically you just very vigorously pipette, and they just start coming up out of their microwells, and then you collect them, spin them down, and then you can reconstitute them in whatever media you need or whatever solution you need to perform your experiments. So this shows looking at the insulin content in some of the islets that we generated using these L-placia plates from Corning. You can see the crimson color of the zinc crystal staining, indicating that there's presence of insulin in the islets that we've generated using this method. And then functionally, besides measuring the insulin content, I've also done some time-lapse imaging, looking at the calcium response of the islets to stimulation by glucose. So when glucose hits the islets before insulin secreted, intracellular calcium levels rise, and then this causes the pulsatile release of insulin from the granules within the beta cells. So here you can see the different glucose. Here's low glucose, 3 millimolar glucose, and you can see that the islets are not as green as they are over time with the presence of high glucose, indicating that we can use calcium as an indication of the function and the response to glucose. And this just shows a graph of that kind of experiment, showing that as we add and expose the islets to high glucose, that over time we can see elevation of intracellular calcium in those islets, indicating that they're responding to the high glucose stimulation. So the other part of the pancreas, the exocrine pancreas, is what we're becoming interested in now, also at Greenstone. And I'll show you why. We have a patient who has pancreatitis who asked us to make their organoids in this approach so that we can get medications to try to find an effective therapy. But so besides making islets, as you go through the different stages of the pancreatic differentiation, basically going to stage 4 here is using the same method that I've already shown you that we use routinely. So once we get to stage 4, then we can start to utilize this approach so that we end up at the end with mature exocrine pancreatic organoids. So this is a publication that I found from King's College London, showing that they did the same approach that we've used to make PDX1 positive pancreatic progenitors. And then they used a similar type of a static organoid production approach. And you can see the ductal organoids here that they've generated using the approach. And in their paper, they used amylase expression as a marker to know that they've made the acinar and ductal cells that they want in their pancreatic exocrine organoid. So here this shows the organoid production at day 14 of the process, when they're PDX1 and KX 6.1 positive, but amylase negative. And then over the course of 5 days, using the media additives that they outline, you can go from this pancreatic progenitor organoid to a ductal acinar cell organoid. And here's the breakdown of the cell population and identity going from day 14 to day 19. Where there's a reduction of the NKX 6.1 positive cells and a dramatic increase in the number of amylase positive cells. Yeah.
Did you also check for LGR5 positive stem cells?
Check.
Observed throughout the time course.
Like
LGR5
undifferentiated stem cells. We've done PCR analysis to see if there's any detectable expression of like OCT3, 4 or other pluripotent stem cell markers, and at late stages, there's no evidence of any remaining undifferentiated cells.
Question. In your previous slide, how much it costs to go to the single blood flow?
How much does it cost?
To
To go from the beginning to the end, the cost is about very like
$5,000.
$5,000 to $10,000.
Okay. And does it generate enough support? Can you generate enough organoid to support one patient?
Yeah, so in my reading, like for cadaver transplants for type 1 diabetes patients, everything that I've read, the estimate is you need like 50,000 islet organoids to deliver the patient. And so in this plate that Corning made, 48K plate, it could make 48,000 organoids at one time. That should be enough to, in one batch, have enough organoids that you could transplant to a patient that would contribute to blood sugar regulation. So yeah, like one production run should be enough. But you know how life is, right? It doesn't always work well the first time. Sometimes you have to do it twice. So it's not that, I don't think it's that expensive compared to the cost of treating type 1 diabetes for 10 years and all the needles and all the consumables and blood sugar monitoring. That those devices work, but I've lived and worked with type 1 diabetic patients, and it's frustrating to like get the amount of insulin correct, even to do the continuous monitoring is like they would be beeping all the time. So yeah, it would be great to not have to do that.
So can I also ask more broadly, I mean, you're giving the example of diabetes, islets, and then exocrine tissue, which is obviously very interesting to me, but can be applicable to many other cell types, and you've got like 2,000 iPSCs stored of different types from various patients with monogenic mutations and so on. I guess my question is a little more broad. Do you find that obviously you're dealing with a progenitor that then differentiates into various cell types, or you can coax it into one more than another. For the islet, for example, can you match the stoichiometry and architecture of the islet? So if you have some delta cells, alpha cells in a proportion that's similar to what you would see relative to beta cells in the human, and then architecturally in the islet, you know, it's not random. They're often surrounding the tissue, and then related to that, I guess. Again, you can't have everything, I guess, but maybe if you could have everything, is islets live in the pancreas for a reason, and when you take the islets out, you do the islet isolation for example, for the total pancreatectomy chronic hepatitis, you infuse into the liver. The islets last maybe for a few years, but there's something like 50% of patients who undergo the intraportal, so installation in the liver, will lose insulin dependence, will become insulin dependent, will lose insulin independence by like 7 or 8 years. And so is it possible to create organoids from iPSCs that mimic the whole architecture, including the exocrine tissue, where even that would
Blood
work?
vessels as well. You need some vascularization.
Yeah, I think vascularization is an important key that we haven't really mastered yet. That's important for long-term survival. Right? So they have to have blood flow, and the blood flow also helps with blood sugar regulation, because then the islets actually see the blood sugar. So vascularization is one thing, and that might require adding in some vascular cells. So there's other protocols that can be used to generate blood vessel cells, endothelial cells, and smooth muscle cells. Maybe if we just sprinkle in those cells with the pancreatic progenitors or the more mature cells when we make the organoids, then maybe there'll be some that might promote vascularization and better integration of the islets.
Use some 3D printed cages to direct specific structure.
Yeah, I'm sure that's happening at Stanford. Also,
Dr. DC1, Dr. Joe DC1 probably will talk to him that there's a possibility that he can generate 3D printed structure. They need to differentiate in them so that they can generate some kind of structure.
Yeah, there's some to create like a mesh, like a scaffold mesh, to where like blood can cross it, but the islets can't get out. So there's also a lot of focus on engineering some kind of mesh or some kind of cage that can be implanted into a patient, and then the islets will basically be protected, but still be able to secrete insulin or glucagon or other hormones.
Different protein. Other than pancreas, what other differentiation program you have at Greenstone?
We have short mixed neurons, for example, so we can make astrocytes. We also make cardiac myocytes, so that's kind of our specialty, is cardiac cells and cardiac organoids, because our founder, Dr. Wu, is the head of the Stanford Cardiovascular Institute, so we have lots of cardiac disease patient cells that we utilize. We also make endothelial cells, pancreas cells, brain cells. We started making kidney, so that's kind of the beauty of the iPS cells, is like the world's your oyster. You can make pretty much any cell type. And now, since it's been, sorry,
gut organoids also.
what kind of ones?
So we are also making gut organoids,
Gut organoids.
like intestine as well.
Yeah, I think the other exciting new kind of product that we have is making macrophages, patient-specific macrophages. So we can also make the inflammatory cells And they can be activated with LPS, and then they start to secrete inflammatory cytokines. I think that's another exciting possibility to study inflammation, but in a patient-specific way. So some laboratories, they'll use like co-culture of cardiac cells to study inflammatory responses. They'll use cardiomyocytes from one source, but macrophages from another person. And some of my friends have told me when they mix them together, the experiment doesn't last long because the macrophages from one person attack the cells of the other person. And so it's a short-lived experiment, which you could have guessed that based on transplant surgeries, right, where there has to be immune suppression. So to be able to make autologous in vitro models where the macrophages and the organoids come from the same patient is, I think, an important development or an important thing to consider. So getting to kind of like also the use of the iPSCs for personalized medicine. So this is kind of like a case study that we're actively working on at Greenstone. So in working with Honnet and with a patient advocacy group called Mission Cure, we were introduced to a family where one of the family's children has chronic pancreatitis. It's chronic, right? Yeah. So the patient is going to the hospital like every 2 months because they have a flare-up of pancreatitis. And the family kind of has gotten fed up, I think, and tired of going to the hospital and then going home. So they contracted us to make iPSCs for their child. And this is the genetic report. So the patient has several different mutations that are known to cause pancreatitis. And the main one we think is the SPINK1 mutation. It's a very pathogenic pancreatitis causing mutation. So we collected blood from this patient, and at Greenstone we generated iPSCs. This is a picture of that patient's induced pluripotent stem cells in culture. And here I made the stem cells into embryonic bodies, showing that they function as they should, and they'll make three-dimensional aggregates. So right now we're actively generating pancreatic progenitors for this patient. We started creating a bank of frozen pancreatic progenitors because we want to enable a workflow to where we can thaw the progenitors and then make organoids like in a batch to batch. Like do some today, do some next week, so that we can do proper experiments with it. And
can we fix the mutation?
Sorry.
Can we fix these mutations in these organoids?
Yeah. The EVs you can freeze. Yeah.
No, fix.
Yeah, we can do that CRISPR editing to fix the mutation.
Do that?
Yeah.
Or have you tried?
Yeah.
Okay. Okay. Great.
Yeah.
In fact, just to be clear, and there's a question, if you had a monogenic, variant, so let's say you've got like 4 here listed, but if you had one, then as a control, do you not try to develop an isogenic control where you correct that particular variant for that particular IPS?
Yeah, definitely. And you can like correct one mutation at a time and see which one is most responsible for the phenotype. If you wanted to find out which genetic mutation is most responsible.
But here the objective was little different, like to find out what drug can fix the responses from these cells, like in terms of high amylase or lipase, like if we are getting after stimulation. So we are not creating isogenic line for this one, but that's more for like R&D purposes if we have to do it.
Does the age of the patient for reaching suppose 50 years old versus 10 years old? Does it difference for
Never makes the iPSCs, not from the blood sample. I've seen if you use a skin biopsy, that's another popular method for iPSC reduction. There's a age dependence, right? Because like someone who's 80, they've had some exposure for 80 years, so the cells in the skin aren't as healthy as from a young patient. But for the PBMCs, we've had very good success regardless of the age of the patient.
Now, but PBMC, what cell in pig? PBMC has lots of cells. Which type of cell type you start from?
We don't separate them. We just isolate the PBMCs.
The whole
And
PBMC.
then the whole PBMCs, and then we reprogram with a virus, a reprogramming virus that introduces the stem cell factors. So we don't know which cell type, which specific type of cell becomes the stem cell.
The reason I'm asking is, some of the different trigger cells, suppose like the big T cell has lots of like rearrangement and some genes are deleted already. So they cannot, means if the genomic fragment is deleted, you cannot go back to the original state. Versus like some cells which doesn't have. Okay, that's a different question.
Yeah, I don't understand it. But we just reprogram them.
One question that I think many of us have is, this is so cool, how do we collaborate with you? You're at Greenstone, and it's a little unusual because you're at a company, but you're like you're closer than most PIs at Stanford.
Yeah, it's easier to collaborate with us than at Stanford, probably.
Just because of distance. Do you recommend a certain method of reaching out to you, or how do you normally establish collaborators? Do you collaborate with? Are you open to collaboration?
Yes, yeah. So we also submit grants sometimes from Greenstone, SBIR grants. We also provide support to academic grant proposals. So if there's a grant proposal that you're submitting and you need to show that you have a source of organoids, we can provide a letter of support, and we can become a part of your grant program also. We can provide like a budget for how much it costs to generate the organoids, the hiPSCs. We can definitely work with you on that. And we also, we have our own drug discovery pipelines, but we also are doing a lot of CRO type work for other companies that don't have access to the stem cells, to stem cell based platforms. So we also can be contracted to do a project. If there's some medication screening you want to look into, we can use our own hiPSC Biobank and find a patient with pancreatitis. You could use this patient that has pancreatitis as a resource to study therapies or mechanisms of disease. Yeah, we can. Yeah, we're very open to collaboration.
Another quick question. So when you use virus to transduce these genes that can cause teratoma, there's a whole outside factor. Have you tried doing those by RNAs like LNPs?
That's great.
Because virus is a dangerous thing. So sometimes like if you don't like that process.
Yeah, that's a great question, and we've been contemplating switching at Greenstone. So the virus method is it's less labor intensive, because basically you put the virus, you just wait. So it's not, so the mRNA requires repeated transfections to do an mRNA based reprogramming, which I've done. But clinically that's more useful, doing the direct mRNA, because there's no virus. There's not even integration in the nucleus, right? The mRNA stays outside the nucleus. So, and also when you use virus, you have to wait until the viral fingerprint disappears. It takes a few passages, but with the mRNA, you can start, right? You can, like, whatever cells are made, you can start using them right away because there's no reprogramming genetic fingerprint that's left.
If you're interested, probably genetically explored. So, she's like here.
Okay. All right, that sounds great.
So how long do they last in vitro, the organoids, for example, the islets? And have you also tested how long they last in vivo if you were to implant them in an image?
So I don't really have answers for the longevity. The organoids we've kept for up to 2 months, but we've always used them within 2 months. So we haven't done just like a how long do they last type of experiment. And in vivo, I haven't done that either in vivo. We have to look to like Vertex and some of the other publications that have implanted them to see what they do. But this is kind of, so now what we're doing for medication screening, so I showed you this organoid plate, it was a flask where you can make 12,000 in one batch. Corning now is made high throughput plates, where in each well, each little tiny well of 96 well plates, you can make 79 organoids per well. So now you have like 79 biological replicates in that one well. And this makes it easier for drug screening because now we can reduce the number of wells that we have to use. Like if there was one organoid per well, then it's like you can't test as many drugs. Here, using this approach where we have 79 organoids per well, we can really increase the number of medications we can test. So for our patient who just wants to know, like, is there a good FDA approved medication that maybe is used for something else that is effective for pancreatitis? Now we can test those drugs in high content and get an answer very fast. And so this is an example. We've done this with cardiac organoids. So we have this high content imager from molecular devices. Here you can see a field of view just in the center. So we're missing some of the organoids, but you can see that from one well you can get a good amount of data from many organoids. And we've tested this once using cardiac organoids. And so here you can see spontaneously contracting cardiac organoids where every time they flash, that's like a heartbeat. So what we want to do is the same kind of approach, but now imagine that they're pancreatic organoids. And now instead of just watching them contract, we're stimulating them with, what are you stimulating with?
CCK.
CCK. So we stimulate with CCK and see calcium levels rise. And then we can use the calcium flux as an indication of the effectiveness of a drug, maybe to see if it dampens the calcium response or makes it less sensitive to the stimulation. And of course, with the pancreatic cells here for the cardiac cells, things are pretty easy because you see the function, it happens like 10 times in a second. But with the pancreas, and this was kind of took me a while to learn, you have to wait for like a half hour. You have to like stimulate and then take continuous recordings because the cellular and organoid response is a much slower time scale for the insulin secretion. And I'm imagining it'll be the same for the acinar cell response. To see it, probably it's going to take more like 30-minute recording instead of 10-second recording. Yeah.
I have a question with the cardiac ones. So these, at least in the IPS space, with the cardiac main concern is like maturity. So I'm just wondering if, like, with this beating pattern that's already like pretty quickly or slowly depending on its drug treatment, how is that normalized versus like a immature cardiomyocyte-based organ?
So these ones, I don't know exactly what the maturation state is. The dogma is that the organoids are going to be more mature, have a more mature structure, more mature function, which may be true. I don't think it's really been tested systematically yet.
Yeah, because I was wondering what the definition of maturity is for like a lot of these CMs, whether it's high mitochondrial density or beating pattern changes or troponin ratios.
I think the troponin ratio is probably the best marker for cardiac maturation. Is the switch to cardiac troponin I from slow skeletal. That's one of the like hallmark, probably the best marker is the troponin I switch.
So I'm going to say it's 1 o'clock, and there may be some really interesting stuff. We have the room for another few minutes, so we'll continue. But for those who need to take off, let's give Todd Herron a round of applause. It's really amazing, and also, you know, obviously Joe Wu's group, so you could directly reach out to Joe or to Todd or any of these folks.
Right, thank you.
Great to have you in the community of SRP.
If you need any, we're a transplant lab and we can do lots of mice, including transplanting islets under the kidney capsule, which is a very quick, easy way of screening them for function.
Mm-hmm.
Do it not by make the diabetic chemically, make the mouse diabetic through chemical intervention like streptozotocin.
Mm-hmm. Well, do you do that here in this building or?
You do it all in there, right here, at the animal house.
Cool. Yeah, that'd be nice to test our immune stealth genetically engineered cell line that we think should be more useful for transplants.
Yeah.
Might be nice to test it in that model.
And you know, just for the exocrine cells, the calcium is actually rather fast.
Really?
Yeah, it's for the pathological calcium signals that emanate from the pancreatic acinar cell, it's within seconds. And the pathological feature is the peak plateau. So you might look for a peak plateau pattern, which is just, I think, within minutes.
So like it goes up and stays high.
Yeah, so it goes up as peak, and then rather than coming back down and oscillating, it'll come up and then
Stays
plateau.
So it's like
It won't
calcium
come all the way
overload.
down to baseline. And the reason is that these crack channels are open. You can actually validate it by giving crack inhibitors to see whether your system can recapitulate, return to normal.
Actually, in the drug list that we have, we are going to test the CM2460, which is a crack inhibitor.
Right.
So that will be validated within that pipeline.
But, you know, the devil's in the details, right? The calcium is just one part of the pathological framework. It's only early signals. Everything that happens afterwards, you're not necessarily going to catch if it's downstream to calcium. So there may be an effect of some of these drugs, but the setup, you may need to look at cell death or some other feature of the acinar exocrine tissue as a proxy.
Right. So the time scale, so you think like reporting every 1 minute would be fast enough, or should we be faster?
I think you have to record more frequently, but the duration of the recording would not be half an hour. It would maybe 10 minutes or 5 minutes.
Okay.
But you have to record more frequently to capture the pattern.
Okay.
Can you hear me on Zoom?
Yeah.
You have a Zoom person that has a question.
Oh, okay, great.
Hey, hi. Thank you so much. That was really interesting. My question is more on the computational side, in the sense that you have, I mean, thousands, potentially dozens of thousands of organoids. And how do you reliably pick out and like quantitate features of those organoids? You know, I think at this scale, obviously you're moving past anything, you know, hand drawn or, you know, anything with the manual annotation. And so there's been, you know, in Nature's come out with a group in Germany on like kind of quantitating using different algorithms and AI and stuff like that. Are you guys kind of going that route? Or I know you mentioned AI, which I think that's where we're all heading. We kind of with like quantitation of these types of things. If there's anything you can say about your quantitation methods would be super helpful.
All I can say is this slide kind of shows that all I can say is that we can collect lots and lots of data, and at the moment we can't analyze it because it's almost too much. It's too much data. So
you said you can't. Here, like you're working on it.
We're working on it, exactly. So you can see here in this experiment where we were using the cardiac organoids, we were doing a 10-second recording per well.
Yeah.
And the size of the data is 2 gigabytes per well. So now you have 100 wells, that's 200 gigabytes. That's about the size of most computers' memory. Is about 200.
Correct.
gigabytes.
I mean, that's where we're now having to use like NVIDIA chips. We have to use AWS. Like you can't use, it's like in order to analyze this much data, you have to, go beyond the personal.
Yeah. So what I'm hoping, so our company Greenstone, one of our lead investors and the chairman of the board is the CEO of Intel. And so now that I have this major computing problem, I'm going to be bugging him and his team at Intel to help us, because if anyone can help us with this computational problem, it should be Intel. So I'm hoping that we're gonna be developing a workstation for analysis of this data. And we're hoping to get one of their fancy new AI computer chips that process data much faster, because it's more based on it's like called neuromorphic AI chip. So it processes data in parallel and it makes data processing faster. So we're hoping that we'll be able to solve this computational problem in our collaboration with Intel. And Shor's leading that effort in our collaboration with Intel. So hopefully hopefully in the next month or two, we'll have a solution to that. But that's a great question. And that's exactly where we are. And that's kind of our pain point at the moment, is processing all of this data.
Same. Okay, thank you. That's helpful. Appreciate it.
Yeah, thanks for asking. You know, I apologize. I think there was a hand up. Maybe you were the hand up. I think if anyone else has a question online, please just feel free to ask it, even informally, as we're wrapping up.
I just had a quick question too. Is Greenstone doing intestinal organoids or?
What kind of organoids?
Intestinal.
Intestinal, yeah. We have a GI organoid expert that we hired in the last year, and we're making also iPS gut organoids, and we also have some patient-derived organoids from biopsy samples at Stanford. So we can do either patient-derived or iPSC derived.
And colon too?
I don't know if you specified colon. I'm sure the Ibrahim can do that.
Okay. My other question was like the drug screening does Greenstone have like in a database of drugs, or like collaborating with like Broad Institute or anything for that, or is just a set of drugs that you guys are using for screening?
Like for the patient that we're trying to find a medication for.
Yeah, do you mean everything, or is it you have a targeted set?
So the patient's family, actually, they produced a list of medicines that, because they're probably more well versed on pancreatitis than certainly than I am, because they're so vested in it. So they have a list of medications that they brought to us, and they were like, we want to start here and work our way down as far as which medications to test. And we're focusing on FDA approved medications because they've already passed the hurdle of safety, and mostly safety.
Thanks.
Yeah, no problem.
For the extract tissue, we do, as you know, we've spoken about this a little bit, but now that I've heard it for the third time, is that if you've got the organoids in sufficient quantity, and we don't need very much, we can run basic physiological tests to see how they respond to, for example, basic stimulation with release of amylase. And the CCK responses that you're referring to are with high concentrations of CCK to mimic what might happen in a pancreatitis scenario. But with lower concentrations, you have physiological secretion of pancreatic enzymes. That's why I was asking about the architecture. I mean, you actually have you looked under EM or under morphology, even electron, or even light microscopy on what the polarization of these cells are?
No,
these are highly polarized cells.
Yeah, we haven't looked at these cells, but for the CCK8, we were planning for both, like do the dose dependent responses to see what dose we should be moving forward for drug discovery.
Using what as your output?
Like physiological, and then we are going to look at few things like calcium signaling, enzymatic secretions, and then also we'll see how the trypsin activity looks in these cells. So these are some of the endpoints we will initially refer to before we start testing out all the drugs. So calcium is just one of the tests we are going to do.
Okay. That sounds good. So when you do amylase secretion, I guess, is it polarized to begin with? Do you have some idea of the polarity of the cell? Do you have markers for polarity?
We haven't looked at it.
Yeah, we still have a lot of characterization.
Do you have precedent for polarity? I mean, for kidney cells, other cells where I mean, that are epithelial in nature, do you establish apical, basolateral?
Not for the organoids.
Not that, so they're not, um, okay, they're non-polar, polarized. Okay. And that could be a limitation
Yes.
because at least, I mean, again, it's possible, like you said, life happens.
Yeah,
Their
that might,
cells, so it's possible that they'll still secrete. But, you know, like, for example, the cancer cells or the, immortalized cells that are like AR42J cells, they have very low secretory capacity
Right.
because they have very low polarization.
Mhm.
So if that's a proxy of what might happen, I don't think you can extract much more than calcium signals. And that too, the calcium signals are so highly polarized.
Right.
That there's this apical, that there's a lateral movement of the calcium. And so maybe a lot more characterization required.
Yeah, so this is the first time we are doing the project with ductal organoids. So we don't know like what level we will get in terms of calcium signaling or release. So those tests initially needs to be done. If we don't see like really good responses in terms of, we can even try different stimuli, but if we don't see responses, then we need to do more R&D on it, like in terms of polarity or like the cell type, all those things that will be tested then.
Very good.
But this is just the first project to start with. I think there will be a lot of modifications going in.
And people have tried, and you showed a paper to do acinar organoids. The problem is no one, to my knowledge, has shown any functional outcome.
No, I think there is one paper from UK or Europe that has done some of the characterization, but not in a lot of details.
So what exactly is the
I need to check. It's been a few months that I
again,
did
the
it.
devils in the details. We had a reviewer for a grant that we haven't gotten the grant for yet. Tell us, you know, that why don't you use organoid? And we're trying to explain to them.
Same comments.
Scour the literature, and there's no functional outcome.
It's the same study section actually. So they may be discussed. Why they don't propose papers. That's why I'm trying to propose this.
No, I think to make reviewers happy, you just say like, okay, we will do it. Then later you can show it's not working, right?
This being recorded.
I mean, that's what I did for my Arwehs. Like he asked for like hiPSC organoids. I said, okay, we will do
Any
it.
resemblance to any characters, life fictitious is entirely coincidental. Yeah.
There you go, exactly. That's a good disclaimer. But I do agree that, I mean, you're doing something that's helpful. Just with the patient, again, I'm thinking out loud here. And I know the patient, we talked about this patient, they're so invested. It's amazing to have someone who's willing to, you know, put in the investment, get a whole company behind it, or a mission cure group. You know, they have their own limitations and resources. Right. But again, the question is, what is it? So let's say you find, yeah, I'm just trying to play it out. So let's say you find you have like 30 drugs, and one of them are crack inhibitors and so on. You see that there's a reduction in the pathological calcium signal. Does that mean that that patient will benefit more than someone else? Or that patient will benefit, period? I guess to start with, the latter question is what you're asking, right? Because you don't have a control per se.
Mhm.
You're not asking whether this patient will specifically benefit compared to someone else more.
Yeah, that's more like R&D thing, like academia R&D. For us, it's more focused on that patient only, like what drug might work for you.
Right, and it might
because
work for everyone, but it might,
that person is like getting 20 or 22 attacks per year. So anything coming out of this project, they might try it out to see if it works, because these are FDA approved safe drugs. So it's like having something rather than nothing, just to try it out, see if it works for or not.
the other issue, again, I agree with you. That's a great start. Calcium is one thing. Again, you're going to set up other things, cell death. I would say trypsin is also early.
Yes.
Like, the field, I think, I mean, I've been in the field for more than 20 years now, and the field is plagued with this hyper, I'd say, myopic view of of pancreatitis as just being calcium, trypsin. But those are, even if they're somewhat universal, they're very early events. What happens afterwards may be as or more important. And for this patient, that might be helpful, because you prevent the attacks and so on. And I'm not discrediting that, but there may be like, that may be 10% of the problem in general. 90% might be what happens afterwards, such as the cell death pathways, fibrosis for chronic pancreatitis. And that too, you may need, that's why I was asking, can you get the parenchymal cells? But if they're from a different lineage, they're not even from endoderm, like pancreatic stellate cells or other macrophages that might lay down some of the fibrotic material. That in the long term may be helpful, but then apart from that, even with the acute, some of the other factors, such as the influx of inflammation, but you could even ask, and this is what, so Steve Pendall had this really landmark paper, I would say, in the JCI from early 2000, 2001 or 2 or something, where he, with the group, showed that the pancreatic acinar cells autonomously secretes cytokines that then can attract other, our chemoattractants. And so you could, within this, look at the secretome, examine whether, or endogenously, whether there's increased expression of cytokine.
Actually, that's one of the plan. We were even thinking to do proteomics on these organoids to see what nodes are being corrected by different drugs, because that will also tell like what can be changed on the line on some of the pathways.
It feels like as you're doing, again, you have a patient, they're motivated, they want to get it started, but the characterization just with the positive control compared to no positive control would be helpful to see how closely it resembles.
Yes, the only problem is positive control in this case, because the patient has so many mutations. It's hard to define even a positive control. Like we are saying that calcium inhibitor will be a good positive control, but we don't know even if that will work for that patient, right?
But then can I ask you, could you just take your own blood or somebody's blood? I'll give you my blood.
No, actually we have the patient's parents' blood who are heterozygous to the mutations.
Right.
But to start with, we are doing only patient's one. And then if needed, we are going to bring in a healthy one, which we are already doing, but also the parents with the heterozygous mutation. So those are our like controls in place for this patient. Like
But yeah, that would be a better experience, but we're also constrained by the cost of it. And the patient, like we told the patient, it would be ideal if we also test the mom and the dad's cells as well as the child's, but the cost was too high. So the customer's like, just show me the patient, show me my sons, and show me the dysfunction, and show me that a drug can modify the dysfunction.
And they are paying out of pocket.
They're paying out of pocket.
It's always restricted.
Wow. I mean, I feel for this patient. I know this patient very well. Thank you.
I think the other arena where this can be useful is for studying GLP-1 drugs, because I think one of the side effects is pancreatitis for GLP-1 drugs. So we'll be probably pitching this to the GLP-1 drug makers as a way to test side effects.
It's not that common. Like it's like those companies are saying it's a rare event. Pancreatitis. They are not agreeing that it's common, but it's a common side effect.
In your experience, is there any genetic basis for the pancreatic response? Because my mom had it.
With GLP-1?
With GLP-1.
Really?
She took whatever the name Monjaro.
Pancreatitis.
Like it didn't move to pancreatitis, but she was having pain in the pancreas. We showed the doctor and she was taken off Monjaro or whatever.
Her amylase went up.
Yeah. So And then I've heard that this is much more common in India than it is here. And I was wondering, and I wanted to ask right after you answered, I wanted to ask you to close down the session.
Pink one is common.
If there's any genetic basis for that pancreatitis response, and that's why it's not common here, because probably,
I haven't looked at the most recent literature,
Yeah.
which is why I was asking the question. But we should put it into cloud code, is that, so I've written a grant a while back, and I tried to scour the literature at the time. There was no, because, before the obesity, even 15 years ago, GLP-1s were marketed for diabetes. And there was this risk of pancreatitis, but now the GLP-1s are longer acting, they're higher dosing. So you can imagine that if it's a dose effect, if it's a longevity effect, that there may be more of a risk. I don't know the answer to that, but that would be interesting. But I wonder whether, so I thought about this.
Yeah.
Is there enough information in a real world data set to ask this question, like the UK Biobank? Are there enough GLP-1 users? Because they have genomics, obviously, available. I don't know the answer.
Yeah. Like calculating a polygenic risk score for GLP-1 mediated pancreatitis.
Exactly.
Yeah.
And
So
they
very interesting question.
Very interesting.
And the UK Biobank is diverse enough for us to get like a population level answer.
So we gave, no, sorry, we didn't, somebody else. There were several people trying to write these grants. Because it's, it's kind of like everyone has an interest in this. I agree with you. I think it's more common than reported. But the studies suggest it's not as common. The question is, can you provoke it somehow? So if you just give GLP-1, people have shown that not much happens to the exocrine tissue, at least ex vivo.
Yeah, interesting. But yeah, thank you for asking that question to SGDS though. It's a very good question.
Is that the guy from Lilly? Definitely want money from Lilly. But the guy from Lilly dismissed everything.
Yeah, they didn't agree to it.
Suddenly everything's okay with all that, and the only problem is that only 1 out of 10 people are taking our drugs, and 9 out of 10 more need to take our drugs. And we have to figure out how to solve this problem.
A societal problem.
Why are you taking the drug on Exactly.